The overall goal of this procedure is to measure the lifespan of C. elegans worms that were treated with drugs of interest. This is achieved by first preparing OP50, a bacterial strain which is used to feed the worms. An age-synchronized population of worms is then generated and seeded into 96 well plates together with the feeding bacteria, OP50. Over the next 2 days, the worms grow to the L4 larval stage and the drug FUDR is added to prevent reproduction. The following day, the worms reach adulthood and the drugs of interest are added to the individual wells. From this point forward, the number of living and dead worms is determined every 2 to 3 days until all worms have died. Observation is done using an inverted microscope, and the number of dead and live worms is recorded in each session.
Hi, I'm Greg Solis. I'm in the Petrushek lab in the Department of Chemical Physiology at the Scripps Research Institute. Today I will be showing you how to synchronize worms and distribute them in 96 well microtiter plates. The main advantage of this technique over existing methods is that it allows for the rapid identification of molecules that extend lifespan in C. elegans. So let's get started.
To prepare feeding bacteria for C. elegans, begin on day -7 by inoculating 5 ml of TB containing 100 micrograms per milliliter ampicillin and 0.1 micrograms per milliliter amphotericin B with a single OP50 colony. Incubate overnight at 37 degrees Celsius in a bacterial shaker. Early the next morning, dilute the overnight culture of OP50 1 in 2000 in 300 ml of TB, containing 50 micrograms per milliliter of ampicillin. Incubate the culture in a bacterial shaker for 8 to 12 hours at 37 degrees Celsius until saturation is reached. Do not allow the culture to grow longer than 14 hours. Transfer the OP50 into a sterile pre-weighted centrifugation tube. Pellet the OP50 by centrifugation for 10 minutes at 3,500 rpm or 2,200 times G in a tabletop centrifuge. Discard the supernatant, resuspend the OP50 pellet in sterile water and repellet by centrifugation. Repeat this wash twice. After the second wash, carefully and thoroughly remove all the remaining water. Weigh the centrifugation tube containing the pellet and subtract the weight of the empty centrifugation tube in order to determine the weight of the pellet. Next, thoroughly resuspend the pellet in S-complete to a concentration of 100 mg per milliliter. No clumps should be left. The concentration of 100 mg per milliliter OP50 should correspond to 2 × 10⁻10 bacteria per milliliter. Use a photospectrometer to determine the number of bacteria per milliliter if the relationship between the optical density and the number of bacteria per milliliter is known. If necessary, adjust the concentration of the OP50 feeding solution to 2 × 10⁻10 bacteria per milliliter. Finally, store the OP50 solution at 4 degrees Celsius until it is used for the worm culture. To generate an age-synchronous population of worms, begin late afternoon on day -6 with a 5 to 10 day old NGM plate on which the worm population consists mainly of starved L1 larvae. Sterilize a metal spatula by shortly heating it over a Bunsen burner. Let it cool. Then use the cooled spatula to cut out agar chunks from the plate containing the starved worms. Transfer several of these chunks onto a fresh 10 cm NGM plate seeded with OP50. Incubate the worms for approximately 65 hours at 20 degrees Celsius until the majority of the worm population consists of gravid adults. The time it takes for the starved L1 animals to grow into gravid adults may vary from strain to strain. After the incubation, collect worms from the 10 cm plate by washing them off the plate with 5 to 10 ml of sterile water. Transfer the worm water solution into a 15 ml conical tube. Wash the worms by centrifuging for 2 minutes in a tabletop centrifuge at 1,200 rpm or 280 times G. Discard the supernatant and add 10 ml of water. Repeat the wash step. Remove the supernatant and add 5 ml of freshly prepared bleach sodium hydroxide solution. Incubate for 5 minutes at RT until the worms break open. Be sure to vortex gently every minute. Monitor the progress of the reaction under the dissecting microscope. As soon as all the adults dissolve, add 5 ml of M9 buffer to neutralize the reaction. Wash the eggs 3 times with 10 ml of M9 buffer by centrifuging for 2 minutes at 2500 rpm or 1100 times G. Then, wash the eggs once with 10 ml of S complete by centrifuging for 2 minutes at 2500 rpm or 1100 times G. Remove the supernatant, add 10 ml of S complete and transfer the solution to a fresh 50 ml conical tube. Add 30 ml of S complete to a final volume of 40 ml. Gently shake the tube overnight at room temperature on a nematode or similar device. To seed the animal plates the next day around noon under a dissecting scope, check whether the worms hatch during the night. Determine the concentration of worms in the S-complete solution by counting the number of worms in 10 microliter drops using a dissecting scope. Count at least 10 drops for each sample. Resuspend the worms at a concentration of 80 to 100 worms per milliliter in S-complete medium. Add carbenicillin to a final concentration of 50 micrograms per milliliter and amphotericin B to a final concentration of 0.1 micrograms per milliliter. Shake on a nematode. If preparing larger quantities of worms, use a 600 ml non-clon flask with a filter cap. At 2:30 p.m, add the OP50 prepared in part 1 to a final concentration of 6 mg per milliliter. Return the OP50 to 4 degrees Celsius. Transfer 120 microliters of the worm OP50 solution into each well of a 96 well plate with the transparent bottom. Make sure to keep the worms in suspension while pipetting. Seal the plate using a tape sealer to avoid contamination and evaporation. Shake the plate on a microtiter plate shaker for 2 minutes and incubate for 2 days at 20 degrees Celsius until the animals reach the L4 stage. Next, to sterilize the animals, add 30 microliters of a 0.6 millimolar FUDR stock solution to each well. This step brings the final volume in each well to 150 microliters and reduces the final concentration of OP50 from 6 mg per milliliter to 5 mg per milliliter. Seal the plate using tape sealers and shake it for 2 to 3 minutes on a microtiter plate shaker. If the OP50 was added at 2:30 on day -2, it is important that the FUDR is added before noon. Return plates to the 20 degrees Celsius incubator. By 9 a.m. the next day, most of the animals should be gravid and contain several eggs each. Add the drugs whose effect on lifespan is to be tested at the desired concentration. If dissolving the drugs in DMSO, the final concentrations of DMSO should not exceed 0.6%, as DMSO concentrations higher than 0.6% shorten C. elegans lifespan. After addition of the drug, seal the plates with tape sealer and shake them for 2 to 3 minutes on a microtiter plate shaker. Return the plates to the 20 degrees Celsius incubator. Adding the drugs can occasionally kill a few animals per plate, especially if using a solvent other than water. Use an inverted microscope to check for dead animals. Generally, there should be less than 10 dead animals per 96 well plate. Return plates to the 20 degrees Celsius incubator for 2 days. To allow fresh oxygen to enter the culture, remove the sealer. Wait 1 minute and reseal the plate. Shake the plate for 2 to 3 minutes on a microtiter plate shaker. Repeat once every week. On day 5 of the adult lifespan, add 5 microliters of the 100 mg per milliliter OP50 solution that was prepared earlier to each of the 96 wells to prevent starvation. Worms are scored for lifespan by their movement 3 times per week from the beginning of the experiment until they die. Use an inverted microscope with a 2x or 2.5x objective to observe worms in 96 well plates. On day 0 at the beginning of the experiment, count the total number of worms in each well. Censor wells that contain more than 18 animals from the analysis, as animals in these wells will not have enough OP50 and will show effects of dietary restriction. In each counting session, record the date and number of animals that move as live animals. Movement in liquid is much easier than on solid media and can be induced by strong lights. The use of a higher magnification may help to spot very subtle movements, like those of the tip of the pharynx. Such subtle movements are often the only movement observed in very old animals. Return the plates to the 20°C incubator. Repeat the counting sessions every 2 to 3 days until all the animals are dead. This is a design of an Excel sheet to record lifespan data. For each well, its coordinate in the plate, strain, drug, concentration of the drug, and the total number of animals alive on day 0 is recorded at the beginning of the experiment. Record the date as well as the number of living and dead animals 3 times a week in order to follow the survival of the various populations in each well. This figure shows results of C. elegans lifespan curves recorded at 20°C and 25°C. The microtiter-based lifespan assay accurately reproduces temperature-dependent changes in lifespan. Similarly, as shown here, the microtiter plate assay reproduces changes in lifespan from mutants reported to have lifespans that differ from wild-type N2 animals. This figure shows how increasing doses of metazapine affect mean lifespan of C. elegans. The data obtained by the microtiter plate assay are quantitative enough to determine dose-response relationships.
Once mastered, this technique can be used to screen large chemical libraries for molecules that extend lifespan in C. elegans. Thanks for watching, and good luck with your experiments.